everyone, today I'll be going through some exam style questions to do with Module 2 about microscopy and cell ultrastructure. These questions are from Physics and Maths Tutor and they are under the OCR specification, but they can still be useful for other exam boards. Please subscribe to help my channel grow and I'll continue to go through the A-level biology specification. This question says, which of the following statements is or are true? Statement 1. Microtubules are part of the 9+2 formation in bacterial flagella. Statement 2. Microtubules can be prevented from functioning by a respiratory inhibitor. Statement 3. Microtubules are involved in moving chromosomes from the equator of the poles of the cell during mitosis. With questions like this, if you're not fully aware of things like respiratory inhibitors or the processes in mitosis, you can try and find which answers seem wrong or which answers you know are wrong and use that to help you narrow down your options. A says 1, 2 and 3 are correct. B says 1 and 2 are correct. C says 2 and 3 are correct. And D says only 1 is correct. Looking at statement 1 to begin with, it says microtubules are part of the 9+2 formation in bacterial flagella. I made a video about prokaryotic cells, and in this video I mentioned that prokaryotic cells, such as bacterial cells, can have flagella sometimes. However, these flagella do not exist in the 9+2 formation the same way that they do in eukaryotes. With this context, you're able to tell that statement 1 can never be correct because bacterial flagella don't have a 9+2 arrangement, so regardless of the presence of microtubules, this wouldn't make sense. Using this, you can cancel out the possibility of any of the answers that contain statement 1 being true as being the correct answer because we know statement 1 is not true. A can't be correct, B can't be correct, and D can't be correct. Luckily, this narrows us down to just C, so we can put C as an answer. In an exam, it's always important to look at what you know and what you don't know to be able to narrow down your options. This question says, which of the following statements describe an organelle which is not membrane bound? A. Contains cristae. B. Modifies and packages proteins. C. Contains digestive enzymes. D. Is made of rRNA and protein. I made a video about eukaryotic cells, and in this video I mentioned which cells are membrane bound and which are not, but this question is going solely off of descriptions. A says contains cristae. We know that mitochondria cells contain cristae, which is the folds of the inner membrane. The mitochondria is a double membrane bound organelle, so we know this is not the answer. B says modifies and packages proteins. We know that the organelle that modifies and packages proteins is the Golgi apparatus, and the Golgi apparatus is membrane bound, so we know this can't be the answer. C says contains digestive enzymes. Well, if we think about an organelle that contains digestive enzymes, we can think about a specific type of vesicle called a lysosome, which does contain digestive enzymes. Vesicles and lysosomes are membrane bound, so this can't be the answer. This leaves us with D, which says is made of rRNA and protein. The organelle itself that is made of rRNA and protein is the nucleolus, which is found inside the nucleus. The nucleolus is not a membrane-bound organelle, and so we can put that the answer is D. This question says, which of the following structures A to D are found in prokaryotes and eukaryotes? A. A cell wall made of peptidoglycan. B. Circular genomic DNA. C. A nucleus surrounded by a nuclear membrane. D. Ribosomes. I made a video going over the similarities and differences between eukaryotic and prokaryotic cells. Going through the options, A says a cell wall made of peptidoglycan. We know that both prokaryotes and eukaryotes can have cell walls, however prokaryotes are the only ones that can have peptidoglycan or murin cell walls. The cell walls found in eukaryotes can be cellulose in plant cells or chitin in fungal cells. So A is not the correct answer. B says circular genomic DNA. Circular genomic DNA, also known as plasmids, are found solely in prokaryotes and are not found in eukaryotes, so B can't be the answer. C says a nucleus surrounded by a nuclear membrane. We can already cancel this out because we know that prokaryotes don't tend to have nuclei anyway. So the answer must be D, and looking at D it says ribosomes, and we know that both prokaryotes and eukaryotes contain ribosomes. This question says, which of the options A to D occur in the nucleus of a cell? A. Synthesis of enzymes. B. Synthesis of RNA. C. Modification of peptides. D. Synthesis of carbohydrates. If you are aware of the process of protein synthesis, you know that first DNA has to become RNA in the nucleus before it can be carried out of the nucleus because DNA itself is too big to leave the nuclear envelope. So we can say that the answer is B because synthesis of RNA does occur in the nucleus in order to allow for the information that is needed for protein synthesis to be able to leave the nucleus. So our answer is B. This question says, the image below is a photomicrograph of a mitochondrion. Part I. State the type of microscope used to produce this image. While looking at the image, it's clear to see that it is a mitochondrion because of its shape and the folds that you can see. Well, the image is black and white, so we can rule out the fact that it could be a light microscope because they produce coloured images. Secondly, the image is in 2D, so we can rule out the fact that it could be a scanning electron microscope. So what we have here is a 2D black and white image. So with that information and having ruled out the other ones, we can say that this image was taken with a transmission electron microscope. When you get a question like this, you can always try to rule out the other possibilities that it could be, or you could use the information that you already know to come to a conclusion. Part II. Identify the structures labelled M and N in the photomicrograph. So, taking a closer look at the image, we can see that M is pointed at an area that's between the folds in the image. We know that fluid exists in the mitochondria that's between the folds and exists in the empty space, and that is called the mitochondrial matrix. So we can say that M is the mitochondrial matrix, or just the matrix for short. As for N, you can see that N is pointed directly on one of the folds. Well, the folds of mitochondria is called the cristae or cristae, so we can put that down as our answer. The mark scheme does allow you to say inner membrane for N because the inner membrane is what folds to make the cristae, but to be safe, I would go with the proper answer of cristae. This question says, sago pondweed is an underwater plant that grows in many regions of the world. Figure 1.1 shows a transmission electron micrograph of a sago pondweed cell. I. Identify the cellular components shown at A and B. So we were informed that this is taken with a transmission electron microscope, so that's why the image is 2D and black and white. Sago pondweed cells are plant cells, so you should keep in mind that it's going to have the same organelles that plant cells have. A is pointing at an area that's white and very big. You can assume that this is a vacuole. This is because vacuoles tend to be light coloured like this. They are large and full of sap, so we can put A as the vacuole. B is pointing at the inside of this area of the cell. You might be tempted to say the nucleus because it's a circular shape that appears in the middle of the cell. However, you should notice that it's inside the nucleus, so we can say that this is the nucleolus. Make sure to pay attention to where it's located and what's around it so you can come to a proper conclusion for your answer. This question says, please refer to Figure 2, which is a photomicrograph of a mammalian blood smear. The smear has been stained with hematoxylin and eosin stain. This is a differential stain. Hematoxylin stains nucleic acid blue. Eosin stains protein, including cytoplasmic proteins, pink or red. I. Explain why it is important to use differential staining when examining a blood smear under a microscope. I made a video about microscopy in which I went into a bit of detail about differential staining. We know that differential staining allows us to see different components of the cell, like the cell itself or its organelles, and it also allows you to improve contrast. This question basically wants you to go into a bit of detail or explain it in context of the image. We can see on the photomicrograph, the red blood cells are pretty clear, and then there are other cells. They are white blood cells. We can say that the staining allows us to differentiate and identify different cells, for example, differentiating the red blood cells from the white blood cells in this case. And we can also say that it's important to identify and see differences between the organelles. In this image, you're able to see the nuclei of some of the white blood cells. Red blood cells are always red, so you can also say that red blood cells would have been visible regardless. This allows us to see other cells like white blood cells, which would have been transparent or colourless otherwise. You should also mention that differential staining allows you to improve the image contrast because otherwise it wouldn't be very clear and you wouldn't be able to tell the cells apart. You can also mention the fact that if you wanted to, you could count the white blood cells in the image because you're able to see each of them individually and so you can differentiate them and count their total. Part II. Using Figure 2 and the information provided, suggest and explain why the cytoplasm of cell C and D reacted differently to the stain. On the image, you can see cell C is a bluish colour and cell D has a red colour while the nucleus looks dark blue. From the question itself, we learnt that hematoxylin and eosin have been used to stain the cells, and hematoxylin stains nucleic acid blue and eosin stains protein pink or red, including cytoplasmic proteins. Using this information, since cell C is really blue, we can assume that cell C has more nucleic acid than cell D, therefore the image appeared blue. You should also say D is more red, which means that it definitely has more protein than cell C. Since the question tells us that hematoxylin stains nucleic acid blue and C appears more blue, not only should we say that it has more nucleic acid, we should also say that because it has more nucleic acid, you can assume that it contains RNA. Do not say DNA, you must say RNA. You can specify which type of RNA, for example, mRNA, tRNA, or rRNA. C and D are both white blood cells, but they clearly contain different concentrations of different things in the cell, so you should mention the fact that different cells have different roles and different levels of activity. Even though these are both white blood cells, they perform different roles in the body and in the immune system. This question says, please refer to Figure 2, which is a photomicrograph of a mammalian blood smear. I. Name the cell labelled B. So you can see that B is pointing at a cell that's red. It has no nucleus and it has somewhat of a biconcave shape. This is a red blood cell. You should say that it's an erythrocyte, which is the scientific name for it. The mark scheme does accept a red blood cell, but I would be more tempted to put erythrocyte as the answer. II. Cells A, C, and D work together to carry out an important function. State the function that cells A, C, and D perform. So we know that this is a mammalian blood smear, and in the blood we don't only have red blood cells. A, D, and C are pointed at cells that have nuclei, some lobed and some are full. We can assume that these are white blood cells, and we know that white blood cells are part of the immune response. So we can put in our answer that the functions that cells A, C, and D perform is the immune response. This question says, the onion plant, Allium cepa, is grown as a food crop around the world. Place ticks in the boxes in the table to indicate whether the statements are true or false. Statement about onion root cells: contains chloroplasts, contains mitochondria, contains 70 S ribosomes in the cytoplasm, have pili, have cellulose cell walls. This is a root cell. Chloroplasts contain chlorophyll, which is what makes places like the stem and the leaves green. The roots are not green, so they don't have chloroplasts, so you can put a tick on false. Statement 2 says contains mitochondria. Well, since an onion cell is a plant cell and a plant cell is a eukaryotic cells generally tend to have mitochondria, which means you can put a tick for true. Contains 70 S ribosomes in the cytoplasm. 70S is a type of ribosome that you can find in the prokaryotic cell. We know that onion cells and onion root cells have to be eukaryotic because plant cells are eukaryotic cells, which means they don't contain 70S ribosomes. Instead, they do contain 80S ribosomes, so you can put a tick on false. Pili is a kind of extension that's found on prokaryotic cells, and since we know that plant cells and so onion root cells are not prokaryotic cells, then we can put the answer as false. Has cellulose cell walls. Onion root cells are plant cells and all plant cells have cellulose cell walls, so the answer is true. Thank you for watching the video. I hope you were able to learn some skills to help you answer the question and learn how to recognize some of the questions and what they ask.